Hello, I'm Sang Lee from Gyeongsang National University in Jinju, Korea. So I'd like to thank the organizers for inviting me to this wonderful workshop with a set of most active network scientists in the world. And I'm really happy to present my network community-based analysis on human chromosome contact networks. So I'll talk about real DNA. I mean, in contrast to Professor Hang Jung's talk on Monday. So there are many precious collaborators of mine who are actually roughly color-coded according to the topics. So first, I will mainly present my work on human chromosome conformation networks analyzed with the network community detection, in particular across different scales. The second part is about methodology on community detection itself, especially on its consistency of the result. And eventually, I'll conclude my talk with an exciting ongoing work, which is basically the combination of the two topics I mentioned. So I'll start to talk about the first work by introducing the human chromosome structure. And as most of you know, chromosomes have this complicated, non-trivial folded structure inside cell nuclei. So when I first learned about this structure in biology class, I believe all of the details about this packing structure is well known. But of course, it was not. And I think this is one of the many examples that are unknown, even if we decoded the human genome sequence itself. And of course, the deciphering this structure is crucial to understand how this fundamental genetic material or chromatin works in cell. And a major breakthrough step to reveal the physical proximity between a genomic sequence is this interaction map of the chromatin loci called Hi-C. So this Hi-C technique provided the physical proximity or the interaction frequency between two chromosome loci in this matrix form. And the technology itself has recently improved enormously for a couple of years. For example, the resolution of the map was in the order of megabase pair in 2009, but it was possible to get the map in the order of kilobase pair in 2014. And of course, you can see some non-trivial structures here and there, depending on the resolution, which is in fact important clues to reveal these 3D structures. For example, this off-diagonal element here in higher resolution is an evidence of the loop structure, like this one, as the title of this paper suggests. Another famous example is in the lower resolution is this block diagonal structure or block substructure called topologically associated domain or TAD, which obviously represent the local packing structure. So, I mean, when I first saw this structure, as any of the audience of this workshop would think, I realized that this is a very familiar problem to me. If we consider the loci as nodes and the weights or physical proximity between them as weighted edges, well, detecting such a block diagonal substructure precisely corresponds to the famous problem of the community detection in network science. So in the first part of my talk, I will present the network community analysis on this chromosome contact network, especially regarding the scale dependence of the community. For details, you can read our first paper published 3 years ago and a recent preprint available on bioRxiv. So a community structure of a network refers to this kind of division of the entire networks into pieces where each of the community is statistically more densely connected inside than the inter-community connections. So this is a fairly intuitive concept, but at the same time, it is quite practical to see the structure and the functional subunit lurking in this entire network system. So one of the most conventional ways to detect such a community structure is to maximize this uh, objective function called modularity. Basically, it quantifies how many of the edges are intra-connected ones compared to the background or expected one expressed by this null model term for a given community partition. I'll come back to this null model part soon in the context of the chromatin interaction. And So maximizing this function corresponds to finding the most densely intra-connected subpart. A bit rather technical but important part you should remember throughout my talk is this resolution parameter, gamma, in front of this null model term. It basically controls the overall scales of the communities found, where larger values of gamma yield smaller communities or the larger number of communities in equivalently, as illustrated here. The problem of maximizing the modularity is, of course, computationally challenging because of the combinatorially exploding numbers of possible divisions here. And in fact, how to cleverly find an approximate solution is the key to this modularity-based technique. As I told you, we need to talk about this normal datum here, especially for this chromosome interaction data, because the product of the degrees or the strength here assumes the only information available to us is the strength or the background interaction for each node. So we use this best guess that the interaction between the two nodes will be proportional to those background interaction. However, in this case, we know much more than that here. And the additional information here is, of course, the fact that chromosomes are basically 3D folded structure from 1D sequence. So it's another nice example of physical networks by Professor Martin Posfai, introduced yesterday. In other words, we can clearly distinguish between the cases of pairs of loci located far away from each other from those very closely located in the genomic or 1D sequence. Moreover, thanks to the studies on this contact probability scaling as a function of the sequence distance, we know that the background interaction between the two loci follows this so-called fractal globule scaling, with the contact probability inversely proportional to the 1D distance. So we use this fractal globule null model term composed of both the overall interaction strength for each node and this scaling behavior depending on the distance in the modularity function throughout this study. For the audience technical enough in this field, we used the version 2.1 of the Jen-Luban algorithm for all the results I present from now on, which is a variant of the famous Luban algorithm, where the nodes are greedily merged to maximize the modularity. And due to the stochastic nature of merging, it is inherently stochastic, which would be the key aspect in the second part of my talk. So I'll just briefly mention the fact here. Before we jump into the real high-CMAP data, we test our community detection algorithm with the various scales on the model structure called conformation-dependent polymerization, or a CDP fractal global model, which is supposedly mimics the chromatin packing with this scaling behavior, the fractal global scaling. With this example, I would like to emphasize the very important advantage of our algorithm compared to the conventional definition of TED, which is the ability to detect the non-contiguous communities, like this segment here, A, B, C, and again A and B. In addition, as the results for different resolution parameters are all independent to each other, the communities across different scales are not necessarily strictly hierarchical, which is in fact the very topic I'll discuss later. In fact, in contrast to ours, many conventional TED detecting methods impose such a restriction, so our method is quite flexible and versatile in terms of the expressional power. I'll show you examples of such a versatility for HiCMap later, but for now, I'll provide some evidence of the validity of our method with this fractal global model. The best part of using this kind of model system is, of course, we actually know the detailed 3D structure because we actually, model it. So let's see if our community detection scheme successfully finds the compact structure of this model polymer system. The plot on the left shows the scaling of the 3D distance versus the 1D or sequence distance inside the detected community. And as you can clearly see here, the 3D or Euclidean end-to-end distance for community boundaries is systematically shorter than the random counterparts. So this clearly distinct scaling behavior for the CDP model's intra-community structure holds for the rescaled 3D structures, uh, rescaled 3D distance divided by the radius of gyration as well. So it means that our community detection indeed identifies loops or at least a compact substructure. In addition to such a scaling behavior, we measure geometrical properties such as the asphericity or relative shape anisotropy. We can check communities we detect for the CDP model are again systematically take more compact or well, literally a globular shape. So based on this validation with the polymer model system, we move to the real high-CMAP data of the chromatin interaction. So we use this particular high-CMAP data introduced in this cell paper with a 100 kilobase pair resolution and converted the raw data to normalized version of interactions with this normalization scheme. By the way, this original cell paper provides the set of the single resolution TED, so we will compare it with our result later, of course. So here's the representative result of the communities we found with our method for three different resolution parameter in the case of the chromosome 1. Here are example of non-contiguous communities here. I mean, these yellow pieces in fact constitute the same community, and so do these light blue ones. And of course, we can detect the communities for every chromosome like this. And the black stripes here, by the way, represent the non-coding region. And as I already introduced, although many communities in different resolution look like being nested, but it's not strictly true, as you can see from this magnified version. And unlike the polymer model system, the real 3D structure of the chromatin interactions is not known. That is what we want to know. So we have to rely on the relative biological factors for verification, of course. And the first signal is this well-known biological marker called CTCF, which is supposedly enriched near the chromatin domain boundaries. So we compare the CTCF enrichment near the community boundaries from our method with the TEDs from the cell paper I mentioned. And if there are significant overlaps, it means that our method can detect the chromatin domain boundaries just like the TED in the cell paper. And of course, in contrast to the single resolution TED, our method can detect the communities across various scales. So depending on the resolution parameter, gamma here, the overlap between the community boundaries and the CTCF enrichment is changed. So as you can see here, for chromosome 1, around gamma value 0.7, the overlap between the community boundary points and both the CTCF enrichments and the TED boundary points reported in the cell paper becomes maximum, measured by this precision value. Therefore, if we adjust the resolution parameter properly, we can recover the TED with the property of the CTCF enrichment. And as you can see here, the number of TED or the non-contiguous pieces of the community per community has the peak around this gamma value close to 0.7 again. So we actually suggest the TEDs are kind of the most fragmented communities, as you can see from the fragmented color here at gamma equal to 0.7. So we successfully recovered the TED structure at this particular range of gamma. Then what about the other ranges of gamma or resolution? One of the earliest findings about the chromatin interaction is this separation between so-called A and B compartment, with not quite creative name, but using this simple PCA reported in this paper. And one of the compartment is more genetically active than the other. So if you detect the communities for very low resolution or using very low values of gamma, can you recover this A-B compartment separation as well? And it's of course possible in principle because this A-B compartment presented in this paper are, non-contiguous checkerboard patterns. And our method can detect such a structure in principle, of course, which is something that cannot be detected with many conventional TED finding method. So here's the evidence of the recovery of A-B compartment from our method. If we tune the parameter values low enough, we obtain two communities, roughly two communities. And as you can see from this overlap here, The largest community from our method roughly corresponds to the B compartment, less active one, and the second largest community roughly corresponds to the A compartment. So if I summarize the results reported in this paper published 3 years ago, by tuning the resolution parameter in the community detection scheme, we could recover previously reported substructures of a high C map. And based on this verification, we suggest this whole spectrum of community scales that may be meaningful for different biological contexts. And recently, in collaboration with my great collaborators in Umeo again, we have re-examined this semi-nested or semi-hierarchical or bow-tie structure of high C communities more deeply. So please check out our recent preprint on bioarchive for detail, but the key aspect is that we quantify how hierarchical or nested these communities are in different scales, inspired by this nestedness metric used in many ecological network studies. So by setting up this mathematical formula to quantify from most nested structure to most segregated relation between the communities in different scales, we examine the level of a hierarchy in this high C community structure. So here, the perfect hierarchical structure would be composed of the community pairs either fully segregated or fully nested structure corresponding to this double peak in the rightmost part and the leftmost part. On the other extreme case, randomly arranged communities would produce a certain distribution of this nestedness measure. So for our high C communities, it's located somewhere in the middle, meaning that, as the title here suggests, the chromosome communities are semi-nested. In other words, it's a kind of mixture of a hierarchical and non-hierarchical structure. And we successfully regenerated these various profiles with the simple hierarchical folding model, starting from the smallest unit of our communities called the domain. And we went a bit deeper in our investigation by classifying the communities into different chromatin states defined in this paper. And roughly speaking here, the regions marked from A to C represent the more active regions than the inactive heterochromatin or D regions here. Basically, our results show that the active regions are systematically more nested than inactive regions. In addition, active regions have higher modularity. So we found that the active chromatin regions are more modular and composed of more nested structure. Now again, please check out this preprint for detail. And of course, the more fundamental biological explanation would be required as future work. And as the second topic today, I'll come back to a bit more fundamental issues of network community detection. And this topic basically comprises the rest of my talk. So the three papers above here present the theoretical issues of a community inconsistency I will describe shortly. And we are currently working on the draft, presenting its application to the chromosome contact network. So let's go back to the square one of the community detection. Well, in fact, Professor Hite Kim very nicely already introduced this concept on Tuesday, but I will remind you of this again. So this kind of statement, detect the community structure, looks like a completely innocent order if you just study network community detection. But if you are a practitioner of the community detection and try to find the community with a stochastic algorithm, you will immediately encounter this situation. So you run this algorithm and you get this very nice result. Well, that's it. I mean, this is the community structure, and you may think that now you can start to write the paper. Unfortunately, if you run this code again and again, you will get these results. Therefore, you will end up with the ensemble of community structure instead of a single result that you can trust 100%. So the issue here is the inconsistency of the result. So is this necessarily a very bad thing that should be fixed? Well, some people think so, but in fact, just like the famous glass is half empty versus glass is half full argument, we can use them actually to study structural property of the networks. First, we can construct this so-called co-occurrence matrix for each node pair, whose elements indicate the fraction of node pairs belonging to the same community in the ensemble of communities found. And if a pair of nodes always belong to the same community or always belongs to the different community, the matrix element will be 1 or 0, respectively. So the intermediate values here indicate the inconsistency. So in this example, only the 3 nodes between the two obvious bulk of communities are responsible for this inconsistency. So based on the co-occurrence matrix in collaboration with Professor Hite Kim and Kentek, we formulated the inconsistency called the companionship inconsistency or COI of individual node by simply symmetrizing it and taking average of over all of the other nodes. So we can treat this COI value as a new kind of a node centrality to quantify the local measure of community inconsistency, which may eventually reveal some functional flexibility of the nodes. Well, if you're familiar with the node centralities, you may ask this kind of a bridginess can be detected with the betweenness centrality as well. That's a very good question. But as Professor Hite Kim already gave you the spoiler on Tuesday, here's our counterexample. Because the betweenness centrality only takes the shortest path between nodes usually, so it cannot detect this bridge part composed of the two consecutive nodes. So we believe that our method, based on the co-occurrence matrix, represent the more genuine type of nodal community inconsistency. And another keen observer would point out that this symmetrization process can be problematic when a node switches between more than two community. And actually, I will come back to the problem later again when I introduce a more sophisticated local measure we developed recently. And as an appropriate example, we generated the clustered Erdős-Renyi random networks where we loosely connected pre-assigned communities composed of the dense random network and put a number of inconsistent nodes intentionally. And the pre-assigned community structure is colored accordingly on the left side. And the shade on the right side corresponds to the COI value. So if we just try to detect the community structure here, the central part composed of those inconsistent nodes is labeled just randomly, but the inconsistency measure effectively capture that the truth. I mean, the membership here in the central part is in fact inconsistent, so we cannot just trust this label at all. And as most of the participants of this workshop would know, this is the most famous or infamous example of the network committee called the Jackary's Karate Club network, where a karate club was split into two communities as a result of the fight between the instructor and the administrator, reported by the sociologist Wayne Jackary in the 70s. And at this particular values of parameter gamma, we found this interesting node with an absolutely larger value of COI than at any other node, meaning that the member is the most neutral about this reported dispute inside. Especially because this node only has a two neighbor, we call this node the outsider. And the second example is the slightly bigger one, the Star Wars characters network. For those of you knowing this stuff, these are networks of characters appearing in the original trilogy of the movie Star Wars, and they are connected if they appear in the same scene. So in this network, the Luke Skywalker, the main protagonist of the entire series, naturally interact with the various characters in different communities, including his father. Well, sorry for the spoiler for this 40-year-old movie, by the way. And of course, he's far from being an outsider, so we call him as a multiplayer, characterized by the large degree and the large COI value simultaneously. So one can set up some ad hoc measures like a product of the degree and COI value to characterize the multiplayer, distinguished from the outsider in the karate club. Just an example. In case you are not comfortable with our result based on the famous or infamous karate club and the fictional characters, Here are more real, real world example networks. For example, if you look at the inconsistency value of this Chilean power grid node, Professor Hitek Kim already introduced on Tuesday, they show more gradual change of KOI across different nodes, which may imply different levels of hierarchical community structure. I'll come back to this exact point at the rest of my talk by elaborating on this. So, in the paper published 3 years ago, we presented the main idea of using local community inconsistency as a kind of a node centrality and its implication, and of course, the correlation between inconsistency and other centrality measures here. But we believe that there should be more systematic studies on this problem. First, we took the resolution parameter here rather arbitrarily, and we didn't have some kind of a global measure to quantify the overall network-wise inconsistency. Thankfully, Daekyung Lee, a smart and outstanding graduate student back then, and now a postdoc in Kentech, read our paper and contacted us for the potential follow-up study. So it was just the perfect timing for us. And Daekyung came up with the global measure of inconsistency called the partition inconsistency, or pi, based on the cutting edge similarity measure for different community partition, and more sophisticated local measure of inconsistency called membership inconsistency, or MAI, both of which has this nice scaling property, namely the effective number of different configuration in the global levels and the independent membership in the local level. So for all the details, please check our recently published paper I'll introduce shortly. Instead of going through all the mathematical terms here is an illustrative example to show the difference between May and Koi. I hope you remember that I said the rather heuristically defined Koi might have some problem with the more than two communities. And here we let the central nodes here switch among three different communities instead of the two communities. Then the newly developed May is good at detecting the bulk part of the inconsistent region, while the Koi is good at detecting even the boundaries of the bulk inconsistent regions. Fundamentally because it is maximum at the regions where the nodes switching between the two communities, even if there are three communities competing each other here in this example. And by far, the most important aspect of this recent work is that we finally started to look at the whole spectrum of communities by adjusting the resolution parameter gamma. So by looking at the global measure of inconsistency, or pi, as a function of the community resolution gamma, we can pinpoint the ranges of gamma where the inconsistency become very small, or in other words, consistent or reliable communities are found, especially when the number of communities stay flat at a certain integer value here, just like the shaded regions here. By using this series of model with the single level and the double level or hierarchical communities, we verified that our method can actually find this most consistent community across different community resolution. And by taking various real-world networks again, we can actually pinpoint the regions of a meaningful community structure. If you recall, our assumption of the hierarchical community structure of a Chilean power grid is verified here by observing this very nice stepping structure of community, stepping structure of a number of communities with the low value of inconsistency throughout this range of gamma. It's not very easy to get this clear-cut evidence in real network, as audience would agree. So for all of the details, please check our recently published paper here. So with this inconsistency analysis, we suggest to pinpoint the most reliable ranges of community scales in this high C map. If you remember now, in the high C map data I mentioned in the first part of my talk. So from the global inconsistency measure on the left, we can see the different chromosomes have a different ranges of gamma corresponding to various levels of reliability. And on the right, we can see the local or node level inconsistency profile across for example, the chromosome 10 here. And again, depending on the chromatin states in terms of activity, we find that the active regions systematically have larger local inconsistency or less stable community. At first, I found this a bit puzzling because we already know that those active regions are more modular and hierarchical. Here, our interpretation is that the more active regions appears to be involved in multiple independent community arrangements, which could induce lower stability, in fact. So, and in contrast, inactive regions are actually tightly compacted within the nucleus, which might lead to some stable 3D co-localization of this DNA segment with higher stability. And in fact, we recently found a paper supporting this argument, and of course, we would need a more fundamental biological basis for our interpretation here as well. And we are currently working on finalizing the analysis and the writing of the manuscript. So yeah, so stay tuned. So here's a summary of my talk. Throughout our journey of analyzing human chromosome contact networks with the network community detection, we encountered various issues such as a tailor-made null model term and comparison to the polymer model, and recently the question of hierarchy or inconsistency. And I believe all of these probably seemingly technical issues could be some keys to open new directions of research. So hopefully the audience enjoy my talk and think about these problems as well. So please feel free to contact me to this email address. And thank you for listening. And I reintroduce all of my precious collaborators and funding source. Thank you.